good afternoon. My name is Yves Pommier. I'm the Chief of the Developmental Therapeutic Branch at the Center for Cancer Research here. It's my great pleasure that Steve accepted the invitation to come and spend the day at the NIH. It's a treat. So Steve Elledge is a professor in the Department of Genetics at Harvard Medical School and the Department of Medicine at Bateman and Women's Hospital. He's also an investigator with the Howard Hughes, and he has been an investigator since 1993. As you have seen on the little flyer, he was born in Paris, not France, not Texas, not Virginia, but Illinois. So he trained initially as a chemist and then moved on to biology, went to MIT, and worked with Graham Walker, where he engaged on DNA with the first transition synthesis polymerase, and then moved on across the country to go to Stanford in biochemistry with Ronald Davis, where he engaged on the DNA damage response, term that we all use now, the DDR, which Steve was really the pioneer for this pathway with the ATR. He moved to Baylor in 1989, and he stayed there for 14 years until Harvard made a good offer to him, and he went to Harvard Medical School. Steve is a member of the National Academy of Science. He's also an American Academy of Art and Science 203, and in 2004 he was elected as a fellow of the American Academy of Microbiology. His latest prize was in December 2016 with the Breakthrough Prize in Life Science. So if you go to his website, you will see what it entails. But it's a pretty prestigious prize, and the website is beautiful. So I would engage you to go there. Steve has moved along across his career, starting from very detailed chemical elements to DNA damage and DNA damage response, and then now engaging in larger data sets. So it's really inspiring to see how much he's moved along and how much he can cover. Thank you for coming. Look forward to your talk.
Great. Well, thank you for that kind introduction, and thank you for the invitation to talk about my work today. So, so basically, I'm going to talk about our work on cancer genomics and understanding how aneuploidy drives cancer. And so we know that when we think about how normal cells evolve into cancer cells, we typically think of the occasional deletion or amplification, but mostly we focus on mutations in genes, driver genes, or silencing or the occasional translocation. But what we don't talk about is aneuploidy. And aneuploidy was really the first hallmark of cancer. It was found over 100 years ago, and nearly all cancers are highly aneuploid. But you know, whether and how aneuploidy drives cancer or is merely a marker of cancer really isn't well understood, or hasn't been for a while. And so what I'm going to talk about today are four things. First, I'll give you an overview of the models of how gene dosage changes, how these changes drive aneuploidy patterns and drive cancer. And then I'll talk a little bit about some new experiments we're doing to look for new drivers of cancer. And these are drivers that can't be identified by mutational signatures, but are present in amplicons. Then I'll talk a little bit about the underlying genetic architecture of cancer drivers that are very tissue-specific, and what that means for cancer gene identification, something called long-tail mutation patterns. And then finally, I'll provide evidence that argues that aneuploidy drives two hallmarks of cancer, cell proliferation and evasion of the immune system. So aneuploidy is not only a marker of cancer, but it's actually actively participating in cancer. So we first got interested in this in 2012, and it came through a very simple sort of RNAi screen that we did on normal human mammary epithelial cells. And we did a very simple experiment. We just asked, well, what genes make, if you knock down these genes, which genes whose knockdown make you grow faster and which ones make you grow slower? And so the genes that make you grow faster when you knock them down, we call them stop genes because they normally slow down the cell cycle, so they're negative. And STOP stands for suppressors of tumorigenesis or proliferation. And then things that make the cells grow slower, we call go genes. So this is very simple. Most of these, because it's a normal cell line, are essential genes, but some of them are actually oncogenes. And the first thing that we noticed is that in the collection of stop genes, we were highly enriched for genes that were known to drive cancer, for known tumor suppressors. And so we thought about, maybe some of these other genes are actually also tumor suppressors in this collection. So that's where we got interested in this. And so we went looking for these, and at the time, a paper had been published by Matt Meyerson's lab that mapped out recurring deletions and amplifications in cancer. And so we compared these gene sets to those to look for genes that might be in these deletion regions that could be functional drivers of cancer. And that led to a whole analysis that brought us to a model shown on the next slide, which we call the cancer gene island model. And what we found in focal deletions is that focal deletions were enriched for these stop genes, the genes that we showed were negative regulators. And they were present about 20% more frequently than you would expect randomly. But even more importantly, we found that they were enriched for those, but they were actually avoiding the go genes. So the frequent focal deletion areas in the cancer genomes are enriched for stop genes, and they avoid essential genes. And they avoid these essential genes at the rate of about 30 to 35%. And we think that this is a signature of haploinsufficiency because the other copy of, these are all hemizygous deletions, so the other copy of the chromosome has all these genes. So you're avoiding the loss of one copy. So we hypothesized at the time that 35% of all human genes are haploinsufficient. And this was in 2012, and since then there's been a lot of support for this, both from the mouse knockout consortium, but also from a recent sequencing of 60,000 genomes, which looked at the distribution of loss of function alleles in genes, and there are about 20% of human genes have fewer stop mutations than you would predict. This is a single copy also, so that's another signature of haploinsufficiency. We then later showed in focal amplifications, you see just the opposite, that you avoid the stop genes, the inhibitor genes, the tumor suppressor genes, and you're enriched for oncogenes. At this point, essential genes don't matter at this point, but oncogenic drivers do. So this is the focal events. What about whole chromosomes or chromosome arm events? And so that led us to do an investigation of these whole arms here, and we built a model that we call the cancer gene dosage balance model, where we measured the likelihood that any particular arm of a chromosome would be more pro-tumorigenic based on the distribution of tumor suppressors and oncogenes along the arms. And if this balance then would make an arm more anti-growth and pro-deletion or more pro-amplification and drive the cancer karyotype. And so we built a mathematical model that actually did this, and we weighted the tumor suppressor genes per arm and divided by the number of genes per arm because there's a certain toxicity, proteotoxic stress, work from Angelica Emon, that we figured was proportional to the size of the chromosome. So we gave it a tumor suppressor gene score for the number and potency of the tumor suppressors, and then we subtract out for deletions the number of the score for oncogenes and essential genes out of this. And so we get a composite score that tells you how tumor suppressor-like that arm is. Then we plotted it against the arm deletion frequency, and what you can see is that we got a pretty good correlation here with a R value of 0.75. And this is for chromosome arm loss, but the same is true for whole chromosomes. And what that suggests is that, so the genes, the tumor suppressors that we're working with now, I forgot to mention, these are not from the shRNA screen. These are derived from a mutation of cancer genomes. So we built an algorithm to predict the tumor suppressor genes and oncogenes from the 8,000 genes and controls that the TCGA sequence. So what this argues is that it's the dosage of the same genes that, when mutated as point mutants, drive cancers. Now it's a cumulative gene dosage sort of model. So that's the background, and most of this is published, so I won't go into it. Now I'd like to talk about our attempts to identify oncogenic drivers that were missed by mutational analysis. And so identifying oncogenes is really difficult. And the way that you do it by sequence is that you look for a pattern, a recurring pattern of mutagenesis, like, for example, RAS, because there are a smaller number of ways that you can actually activate a protein as opposed to inactivate it. But some genes or some proteins are very difficult to activate by point mutations, but if you increase their dosage, you can get a gain-of-function phenotype. And so, so in order to look for these sorts of genes, we went to doing more genetic screens. And what we did was that we built an open reading frame library of about 30,000 sequence-verified ORFs that represent about 16,000 genes. And we barcoded these, and we put them under an inducible promoter. And this just shows you when you have GFP in this promoter, it's a TET inducible promoter. If you add the TET analog doxycycline, you can induce the expression. And then we put them into normal cells. So we put them into human mammary epithelial cells. These are immortalized with telomerase, pancreas-derived cells, or fibroblasts. And then we just add dox or don't, and we let them grow for 10 generations and look to see which genes enrich and which genes make these cells not proliferate. And so this is what the data actually looks like. You can see that we get enrichment of some genes. We call these go genes because they're promoting growth. And we see the dropout of a lot of genes. We call these stop genes. When you look at the types of genes that are driving the cell cycle and proliferation, there are a lot of familiar faces like MYC, CDK6, D-type cyclins, receptor tyrosine kinases, things like that. Of course, there are a whole lot of genes that we've never heard of before, and that was one of the big surprises. And things that drop out, a lot of things you might expect, the CDK inhibitors shown here on the lower right, but also interferon genes. We know interferon expression slows cells and makes them sick. And when you map the positions of these go genes onto a map of chromosomes where the amplicons are shown, you can see that, that we actually fill in a lot of gaps. We found almost 150 pro-proliferation genes in recurring amplicons. And the recurring amplicons that are mapped here are either pan-cancer, so they're in a lot of cancers, or they're specifically in breast or pancreatic cancers. We didn't do sarcomas that the fibroblasts would do, but you can see that we found a lot of potential drivers. Now, we don't know how many of these drivers are functional, but we know that we found 50% or greater enrichment for these genes in those amplicon regions than you would expect randomly. So we think we have some drivers. So now I want to talk about how these drivers behave in different cell types. Because there we found something quite surprising to us. And as I said, we did 3 normal cell lines. And so what we found were that the 2 epithelial lines, the breast and pancreatic lines, had the most similar pattern of genes relative to fibroblasts. So that makes sense at some level. And when you look at the overlap between them, what you can see here on the right, if you focus on the stop genes, there's a lot of overlap. Depending on which side you look at, it's between 40 and 63% overlap. And there are a lot of genes that can make cells grow slower. This is all the same statistical cutoff of a p-value of 0.01. And so there are a lot of ways to turn off cell proliferation and make it not work well. But the big surprise were the genes that made cells proliferate. And there we got a very small overlap. And we thought that would be something that would be more conserved. But we only got about a 10% overlap between these genes. And so that was really surprising to us. And not only did we not see that much overlap, actually the reverse overlaps between the go genes for HMEX and versus the stop genes for HP&E, they had almost the same overlap, which is shown here in the left. Here are the go genes. It's almost the same percentage, a little bit less than when you compare the go versus go genes. And the same is true over here for the opposite comparison. So, and if you look at all three of the cell types that we looked at, and we have two different libraries, so this is only one of the libraries, so the number's a little smaller, but what you can see is that the overlap between all three of these cell lines is there are only 3 genes that are in common. So this suggested to us that these cells are really wired differently, that they're going to respond very differently to different types of growth signaling. And so that was really the big take-home from that. The other thing that we found that was interesting related to the kinds of genes in these libraries that are known to be mutated in these tumors. So we looked at common oncogenes and tumor suppressors and how they behaved. And in this graph, we just have on this we have the HP&E pancreatic cells, and it gives the p-value of whether they drop out or enriched. And the same thing's true for the mammary epithelial cells. If they're enriched, they go up here. If they drop out, they go down here. And so you can see that there are a lot of common drivers that drive cancer, act like go genes, and there are genes that are known to inhibit proliferation in common. Now, if you look specifically at breast, what you see is that the oncogenes that drive breast cancer, MDM4 and the PGF receptor, score well for HMEX, but they do virtually nothing in pancreatic cells. Likewise, these 3 tumor suppressors in breast, when you overproduce them, they drop out of these HMEX, but they have no effect at all on HP&E cells. And the same is true for the tumor suppressors that are specific to pancreatic cancer, pancreatic adenomas, the oncogenes and tumor suppressors. You can see the oncogenes like KRAS and BRAF, they stimulate these cells to proliferate, but they do nothing in breast cancer, and they're also never mutated in breast cancer. Likewise, tumor suppressor, TGF-beta receptor, does nothing in breast, but it drops out of these pancreatic cells. So we can look at the groups of genes that function in these different cell types and plot them based on either things that work in both of these cell lines, the mammary and the pancreatic cell lines. These are basic CDK inhibitors and MYC and the Wnt pathway. But when you look at this, you start to see that there are groups of genes that function only in one cell type. These were gene ontology categories that we found. One are genes specifically involved in pancreatic beta cell development, specifically showed up in the pancreatic cells. Also sets of pancreas-specific transcription factors and nuclear receptors. And in breast, we see prostanoid ligand receptors, which you might expect, but also breast-specific transcription factors. And I want to point out in particular a group of breast-specific proteins called keratin-associated proteins. And these are actually also normally expressed in the skin that cross-link keratins. And there's 2 families, one of which is very cysteine-rich. And this is how this whole family of genes, there's dozens of these genes, how they behaved in log fold 2 enrichment in the breast cancer screen. And you can see certain families, all the members of these families make you proliferate, and there are other family members that don't at all. Now, if you look and say, well, how do they behave relative to other gene sets? Well, if you compare it in mammary cells, olfactory receptors are neutral, but virtually every one of these are either positive or have no effect at all. And if you look at how they behave in the pancreatic cells or in the fibroblasts, they basically have a neutral effect. So they're really specific for stimulating these normal breast cells. We were interested in whether or not they might be overexpressed in cancer in general, so we did a comparison of the two different types of these KTRAPs, the neutrals and the GO genes. And you can see that for the neutral genes, if you look in the normal tissue or the tumor tissue, there's virtually no difference. But for the GO genes, you often see wherever these asterisks are that their expression is enriched in the tumors from these different tumor types. So they appear to have some potential, at least, to be expressed highly in tumors, and they may help drive these tumors, at least potentially to this correlation. So I think 7 of the 11 tumors that we looked at here statistically overexpress these in tumors specifically. So we wanted to know a little bit more about how some of these tumor-specific drivers might work. So we took cells, these are now HMEC cells, and we overproduced them and looked at their transcriptional profiles to see how they behaved. And this is just a principal components analysis shown over here using the transcriptome data. And what you can see is that this is EV is an empty vector. And D-type cyclins and the KTRAPs localize together, and they're farther away from loss of function of P53, SARC expression, or MYC expression. So these kind of look more similar. If you look at do sort of clustering of gene expression, what you can see is that the KTRPs and cyclin Ds have a similar signature, where they overexpress cell cycle genes and protein synthesis genes. And this set is also conserved with MYC, but not any of the other MYC signatures are showing up here. And they're quite different from these other cancers. So you can see they cluster together. So cyclin D and K-traps look very similar. What we know about how cyclin D and drives a cell cycle is, if you look down here, D-type cyclins inactivate a tumor suppressor called RB, and RB inactivates in this complex a transcription factor called E2F that, after cyclin D phosphorylates it activates the G1 to S transition. And so we looked at E2F1 RNA levels, and what you can see is that they increase when you overexpress KTRAP by over 2-fold, and this is p-value here, is quite high for this. So we think that part of this signature may be because KTRAPs are driving entry into the cell cycle in a way similar to D-type cyclins. And this is new data, so we haven't gone and done the epistasis analysis and to look at this in more detail, but we think that there's probably a link here. And this link should be specific to breast cells, and we don't understand that, but we think it's very likely to be the case. So the other thing that we started looking at is how these genes behaved in amplicons in different types of cancer. So if you look at the GO genes that were found in the HMEC screen or the pancreatic cell screen, they're both enriched as a percentile in the pan-cancer amplicons, 50 to 60% more than you would expect. But if you then look in amplicons that are specific to breast, what you see is that HMEC GO genes are even more enriched in the breast amplicons, but the HPNEs aren't. And the opposite is true if you now look at the amplicons in pancreatic cells. Now you see that the GO genes from the HPNE screen are much more enriched than the HMECs, the GO genes. So there's some specificity now, not only in vitro, but in real cancers. And the same is actually true of the stop genes, now looking at recurring deletions in these. So in the pan-cancer, you see more of these stop genes in these recurring deletions, the HPNEs look a lot better for whatever reason in the pan cancer. But you can see if you now look at deletions specifically in the breast, the stop genes are even more enriched if they were found in HMECs than you see if they were found in HPNEs. And the opposite is true if you now look at recurring deletions in pancreatic tumors. Now the HPNE stop genes are enriched. So we also asked the question, excuse me, we also asked the question now, so these are different cell types. If you look at tumor types, if you look at tumors that are more related to, say, breast tumors or more related to pancreatic cancers, does this pattern hold? And so we looked first at ovarian and uterine cancers that are more similar to breast cancers because they're cancers of the female reproductive system. And what you can see is that the Go genes now from the HMEC screens, the breast screens, are more enriched in ovarian amplicons than are those identified in the pancreatic screen. And the same is true for the amplicons found in uterine cancer. The genes that make the breast cells grow better are more enriched in those than the genes that make pancreatic cells grow better. If you now switch and look at tumor types that are more related to pancreatic cancer, now you get just the opposite, that the breast Go genes don't do much at all here in stomach or colorectal cancer, but the Go genes from the pancreatic cells now are enriched. So this suggests that that the amplicons that you see, so if you look at amplicons in different tumor types, you see some that are conserved across all tumors. These are the pan-cancer amplicons. And then you see some that are specific to different types of cancer. And so one of the arguments about how these things work is that it's a chromatin environment's different, and so you just have a higher frequency of certain regions that are amplified or deleted in cancers based on the structure of the genomes, which differ from cell type to cell type. The alternative argument is that different genes are being selected for once these events occur. They occur at a somewhat even frequency, but then selection acts on it. And if that's the case, then if these tissues are wired more similarly in terms of what genes they respond to, you would expect that their amplicons would be more related if the tissues were related. So we looked at recurring amplifications and deletions across a number of different tumor types, and then we did a unsupervised hierarchical clustering, and what you can see is that the amplicons actually predict the relatedness of the tissues during development. So the breast tissues cluster with the uterine and ovarian cancers based on their amplicons, and the pancreatic cancers amplicons cluster with stomach and colorectal. And we also see melanoma and gliomas and glioblastoma cluster together, and they're all derived from the neuroectoderm. And so, so basically, what this argues is that the wiring of all these different tissues determines what sets of genes are able to influence their proliferation and tumorigenesis, and that these are driving the patterns of amplification and deletion that are specific to different tumor types. So now, in the last few minutes of this talk, I'd like to talk about how aneuploidy actually is driving cancer, and what the impact of aneuploidy is on cancer. We've talked about the different genes that are affected during amplification and deletion of chromosomes and chromosome arms, or focal events, which are less than a chromosome arm. But how that actually works, what it actually does to promote cancer is what we're interested in. And so once we started giving scores to aneuploidy and trying to predict which chromosome arms were deleted or amplified in cancers, we realized that we could actually develop a scoring matrix to actually give a particular tumor an aneuploidy-like score based on the number of chromosome alterations, chromosome arm alterations, and focal events. And so we built an algorithm or a mathematical model to give a score for somatic copy number alterations for every tumor. This is just a heat map to show you that certain chromosome arms across all cancers are recurring, or certain arms like to be lost. It's a heat map, or gained, shown in green. So we did this, and this allowed us to then separate tumors based on their aneuploidy score. And so then we took the top 30% of most aneuploid tumors and the bottom 30% of the least aneuploid tumors, and we looked at the transcriptomes of those tumors. And then we started looking for gene sets that were enriched in tumors with high aneuploidy. And so this is a gene set enrichment analysis called GSEA. And for those of you who aren't familiar with it, the way this works is if you look at this heat map down here, high to low, we compare every gene for the difference between high aneuploidy and low aneuploidy, and then we plot its ratio from the highest differentially expressing in the high aneuploidy to the least, so they're most expressed in low aneuploidy. Okay, and that's for all the genes in the whole transcriptome. And then you take gene sets and you map them onto this. And if you take a neutral gene set and map it onto each one of these black bars is a gene, then you get if it's a random gene set or an uninformative gene set, it just has an even distribution, and you basically get a straight line across. But in this case, we looked at the DNA replication genes, and what you can see is that they're most highly expressed in the high aneuploidy tumors. And this is true for cell cycle mitotic genes or DNA replication, and these are all cell cycle regulated genes, transcripts. And so what that argues is that high aneuploidy tumors are more highly mitotic. They give a enrichment score for this and all that, but basically you can just see by eye that they're all piling up to one side. And so this makes sense because a lot of the genes that are driving aneuploidy and that are enriched in aneuploidy are these genes that affect the cell cycle. So, and this had actually been seen previously in data in cell lines in vitro, but this is, I think, the first time that anyone had seen it in tumor cell types. But so that makes sense. High aneuploidy is selected for. You're selecting for a higher frequency of mitosis and cell cycle, and that drives cancer. But the other thing that we found that was surprising, that wasn't known previously, was that we found that there was a gene set that was actually depleted in high aneuploidy tumors, and these were involved in the immune cell markers. Now, tumor cells don't express these immune cell markers, of course. They're specific to immune cells. But when you take a whole tumor and sequence it, you take everything in the tumor, and that includes cells that infiltrate the tumor, including the immune cells. And so if you look at the plots here for GSEA, what you can see is that all these gene sets for either CD8 and natural killer cells or interferon gamma signaling, they're all over on the right. So what that says is that low aneuploidy tumors have a higher immune infiltrate than high aneuploidy tumors. So what was previously known about the immune infiltrate in tumors was a paper by Nir Hacohen's lab who showed that the number of of mutations or neoepitopes in a particular tumor positively correlated with immune infiltration. And that makes sense. The more mutated you are, the more likely you'll be noticed by the immune system. And for 3 tumors here, these are all different tumor types, but these are colorectal cancers, these are lung adenomas, and these are uterine and endometrial cancers. You can see that there's a positive correlation. Now, the p-value, the correlation coefficient is pretty weak. It's about 0.2, and the p-values are just barely significant, 10 to the minus 2, 10 to the minus 3, but there is this positive correlation. But, and just pay attention to these 3 with the asterisks, because I'm going to show you in contrast what it looks like for this correlation with aneuploidy. And that's shown here. So if you do this correlation now, instead of the mutation number, you do it with the aneuploidy scores, you see that you get a negative correlation. The higher the aneuploidy score, the lower the immune infiltrate score for these 3. So now instead of 0.2, we have correlations almost to 0.4, and the p-values are between 10 to the minus 6 and 10 to the minus 9. And some of these are even stronger. Head and neck, the correlation coefficient is 0.45, and the p-value is 10 to the minus 16. And the only 2 tumor types that don't show this are brain cancers. This is low-grade glioma and glioblastoma multiforme. They actually have negative correlation there. So, or positive correlation over here for the low-grade gliomas. So for all the epithelial cancers, we see a strong correlation. This is just another way to look at it. And so what I'm plotting here is just a heat map of the false discovery rates for these. So 0.01 is a very low false discovery rate. You can see just by looking at the colors that the brain cancers don't follow these rules, but all the other cancers do. Ovarian, stomach adenoma, melanoma, kidney cancer, breast cancer, and colorectal cancers. These are the gene ontology categories, and they're kind of hard to understand, but let me just break it down in terms of cell types, because we built up a cell type specific signature. And what you can see is that the CD8 T cell effector cells show a significantly reduced level in high aneuploidy tumors, as do B cells, natural killer cells, and also T regulatory cells. Now, T regulatory cells are negative regulators of the immune system, so you might think that goes in the wrong direction, but I just want to point out that although they're depleted, they're not depleted nearly as well. So you can see that the false discovery rate is much higher for these. But the ratio of Tregs and CD8 cells to Tregs is very significant. So what little immune environment is left in these tumors with high aneuploidy, it's even more anti-immunogenic. So, so now that we've identified these 2 different gene sets, the cell cycle gene sets and the immune infiltrate gene sets, we can ask more specific questions like what aspects of aneuploidy actually drive these 2 different responses. So the way that you can do this is that we're taking advantage of a mathematical method called lasso, which is sort of a machine learning method where you can feed parameters into it and ask them to weigh the parameters and say, how best can you weight this parameter to predict this particular outcome, like low aneuploidy or high cell cycle gene expression? And so you feed it in different parameters, like different types of aneuploidy, and then you ask for it to weight those. And it gives you a beta coefficient, which tells you how much to weight it. And a lot of times, if you put in parameters, it'll just give you 0. They don't predict anything at all. But when they have a positive prediction, it'll give you a beta parameter, and that can be either positive or negative, depending on whether or not it is predictive or anti-predictive. And And so the parameters we put into this particular analysis were focal somatic copy number alterations or whole chromosome and arm alterations. And if you look at the top here, the cell cycle signature, it gave beta coefficients for both of these that were positive, and the focal coefficient is higher than the coefficient for the arm. So that means they're both positive, and it means focal events actually have a more potent ability to predict this immune signature, and then whole chromosomes and arms, and that makes a certain amount of sense because when you think about a recurring focal event, you're basically picking out the sort of sweet spot of the genome that is most enriched for the genes that you're interested in and avoiding the genes you don't want there. And if you then take the whole chromosome arm, you're sort of taking the good with the bad. So it should be less potent of an event and therefore be selected less efficiently. And so and be less predictive. And so that's exactly what you see here. Now, if you look at the immune signature, we get something that was really unexpected. What you can see here is that now the focal events have very little predictive value, but the whole chromosome and whole arm events are very potent. And so different aspects of aneuploidy are predicting the two different signatures. And if you look at, instead of for pan-cancer, if you look across all cancers, what you can see is that you pretty much see that the focal events are the strongest up here, but there's a lot in this heat map, there's a lot of arm events and weighting of arm events. But for an immune signature, it's almost all whole chromosome and whole arm events. The exceptions, of course, are glioblastoma that goes in the wrong direction. But all the epithelial cancers look this way. And so I think what this argues is that there are fundamentally different mechanisms that are driving these two different signatures. The first one is driven by focal events and genes, particular sets of genes. But the second one likely is a property of the whole chromosome and not particular gene sets. Now, I'm not saying that genes don't drive it. We know that's not true. Genes drive immune evasion. There's no question about it. But we think that whole chromosomes do too, and that's probably some property that's unique to the proteotoxic stress that is due to the imbalance of complexes across the proteome. Because when you make an entire chromosome, a lot of the genes are made at the wrong, or proteins are made at the wrong levels. They end up being turned over, and that generates proteotoxic stress. We also fed into the lasso a lot of other parameters, and this pretty much held for the immune infiltrate. We put in the number of mutations and a various number of other parameters, tumor stage, age, and gender. And what we found, again, was that the main predictor is chromosome arms. Occasionally, you'd see focal events, but it's always of lower magnitude. And also, the number of mutations often had a positive score, but again, the absolute value of that is always less than the chromosome arm scores shown here. So we think that aneuploidy is actually driving this, but driving the signature, but it is just a correlation. So in order to try to test that, whether it actually was driving, we went to a mouse cancer model. First we did a human cell analysis. We'll get to the mouse in a minute, where we took these colonic epithelial cancer cells that were lacking P53, and these are primarily diploid cells. So we wanted to make isogenic aneuploid cells out of these. So we transiently inhibited the mitotic checkpoint using spindle checkpoint inhibitor or siRNAs to a spindle checkpoint gene, MAD2. And what you can see is that the chromosome number now goes from 46 to a lot of different aneuploid numbers, both loss and gain. And then we asked whether or not T cells could productively interact with these. So each of these, the parent cell was expressing a precursor protein for a protein that generates a T cell epitope. And we had this CD8 T cell, this is an HIV protein, that was specific for this epitope. And you can see that in the aneuploid cells, they express the same levels. And then we looked at CD8-mediated cell killing. So if we add a nonspecific T cell, the diploid and aneuploid cells look about the same. And this is whether they're treated with control or reversing or siRNA MAD2 or scrambled. And what you can see is that the diploid cells, now if you add the T cell that's specific, this is killing, fraction of killing. They're killed very efficiently. But the aneuploid cells now have a cell autonomous defect in killing. And so we think that not only does aneuploidy correlate with immune infiltration, but it actually is impairing the immune response. We also went to a mouse model where we took a tumor that evolved in a black 6 background, and we made it was essentially diploid. We made it aneuploid, and you can look at how they grow in culture. The diploid cells grow a lot better than the aneuploid cells because we know aneuploid cells are sicker, and a lot of work from Angelica Amon's lab has shown this. Then we took these tumor lines, the diploid and aneuploid version, and we put them into a wild-type mouse or RAG-deficient mouse. And what you can see here is how they grow in the RAG-deficient mouse. The diploid cells grow a lot better than the aneuploid lines, just like they did in vitro. So the tumor volume grows much more slowly. But then we put them into a wild-type mouse that had a wild-type immune system. And now you can see that these two tumors now grow about the same rate. And so the wild-type or the diploid tumors take a much bigger hit than the aneuploid tumors. And when you take these tumors out and look at the immune infiltration, looking at infiltrating T cells, you can see the diploids have more than the aneuploid cells. These are all CD8 T cells. So this is recapitulating what we thought was happening in vitro is now happening in vivo. So we think that these things are really causal. And what's the significance of this? Ability to escape the immune system. We asked whether or not aneuploidy can potentially impact therapies such as immune checkpoint blockade. So we looked at patients who'd been treated with CTLA-4 in 2 different clinical trials where they looked at the genomes of the tumors for these patients. And what you can see is if you just divide these tumors now into the top and lower 50% of aneuploidy and then plot their survival in response to anti-CTLA-4 treatment, what you can see is that the high aneuploidy tumors patients don't respond nearly as well as the low aneuploid tumors. This is from the Van Allen study in Levi Garraway's lab. And in a smaller study, Snyder et al., we get a very similar response. If we look now what the mutational burden does, it's just the opposite. If you have high mutation load, you're slightly better at responding, but it doesn't reach statistical significance, unlike aneuploidy. And if you add these together, you get an even better score. So it looks like they're largely acting independently of each other, and that's true in both of these sets. Let me just summarize what I've told you today. That first of all, that we think that the balance between these growth-promoting GO genes and the growth-inhibitory stop genes, which are tumor suppressor-like, along the chromosomes ultimately determines the selective pressure that drives somatic copy number variations that you see in cancers. And this is all due to this sort of cumulative gene dosage effect of multiple different genes that have small effects, but collectively they actually have a role. The other point I wanted to make is that in different cell types there's an underlying epigenetic or genetic architecture or regulatory architecture that controls proliferation responsiveness and ultimately the patterns of somatic copy number alterations in tumors. I gave you presented evidence that aneuploidy drives two hallmarks of cancer, proliferation and immune evasion. High aneuploidy promotes cell proliferation but inhibits immune infiltration, and the opposite is true of low aneuploidy. And what's interesting about these is that the proliferation signature is predicted by focal events greater than arm events, but the immune evasion is predicted largely by whole chromosome and whole chromosome arm events. And we think that that's a distinct mechanism, and we can talk in the question and answers about what that is. And finally, that aneuploidy is a much stronger predictor than mutation load for immune infiltration, and it better predicts the outcome of immunotherapy. And this is in trials just for CTLA-4, and it remains to be seen if this was also going to be the case for immunotherapy with anti-PD-1 antibodies, and we're eager to do those analyses. So let me just stop and thank the people who did this work. The initial work on the Cancer Gene Island was work from Nicole Solomini, and we had help from Craig Mermel and Ramin Barokam in Matt Meyerson's lab and doing the analysis of that. The work on the Orpheome Library overproduction proliferation screen was work of Laura Sack, and she also collaborated with Teresa Davoli on this. And also the work on the mathematical models for gene balances along whole chromosomes arms that drive the aneuploidy patterns was work from Teresa Davoli in collaboration with Peter Park's lab and Andrew Hsu in his lab. And also the work on the correlations with the ability of aneuploidy to drive cell proliferation signatures and immune evasion signatures was spearheaded by Teresa Davoli with help from Hajime Uno and Dana-Farber, our mathematical collaborator. So thank you for your attention. I'm happy to take any questions.
Hi, Steve. Very nice talk.
Thank you.
So how do you think the immune system recognizes whether there's chromosome imbalance?
I think that the implication is that that the T cells are not recognizing the tumor as well. Certainly, they don't kill cells as well, at least in the model systems that we've developed. And the idea is that in order to get tumors infiltrated by the immune system, that it's sort of a forward-feeding loop, that if the T cells get in and kill cells, those cells then get presented to antigen-presenting cells like dendritic cells. They then stimulate more T cells to proliferate and become active, and then they go back in and kill more cells, and it's a positive feeding loop. If you can dampen that at any stage, you'll reduce the immune infiltration. So we think there's going to be an issue with antigen presentation. One idea is that and by no means the only possible explanation, is that now you're making the wrong numbers of a lot of wild-type proteins, and they're sort of flooding the proteasome and being presented, and therefore the percentage of neoepitopes that get presented is reduced because they're being outcompeted to some degree. Okay. And I think even a 50% reduction is likely to have a significant effect. And the reason for that is that there's a way to reduce presentation by 50% by mutating beta-2 microglobulin, which is the partner for MHC. And so for all MHCs of class 1, and if you get rid of half of that, that's selected for as a mutant in cancer all the time. So that's an argument.
Other quick question: you had a difference in growth between when
Oh,
primary.
sorry. Say
Just when primary cells were aneuploid, they grew poorly, whereas the tumor cells grew much better if they were aneuploid.
They didn't grow better; they grew worse.
The tumor cells, there was a growth advantage
Relative to diploid.
Yeah. Whereas primary cells, there was a growth disadvantage.
Right.
How?
So why did that work?
Why is that?
Because I think there's two variables that are varying. So one is their intrinsic ability to duplicate, and the aneuploid cells are really bad, right? And so the diploid cells are just outgrowing them. But now you lay on top of that that the diploid cells are just being killed a lot by the wild-type immune system. And so that reduces them down to about the same level. So they're probably still proliferating better, but they're getting killed by the immune system, and then they sort of converge. So I think that's probably explains it. This is at least the way we think about that.
I'm assuming that most of the studies you showed were done on human cells and human cancer lines. Is that so?
Yes, that's all the statistical analyses, except for that one mouse study at the end.
Okay, the one mouse study, keep in mind that they're telomerase expressors. Therefore, they're not going to necessarily reflect what's in human. My question is, there's two models. One is a naked mole rat, and the other is a blind mole rat. Both are mammals who are the exception to the rule that they do not develop cancer. And they both live for an extremely long time, about 30 years or so in comparative. And they have unique methods of tumor suppression. Have you ever thought of putting within your analysis how they're doing it and do it on a weighted relative analysis with what you have?
I don't think we have the necessary data on that to do the analysis, but you could potentially do cell line studies. They did recently find 2 cancers in the naked mole rat, so they found 2 different cancers in the naked mole rat. Mole rat recently. So they do get some cancers. But I think that's interesting. Whether or not the reason they have fewer cancers has to do with, a better immune system, I think that's an open question. But we're not in a position right now to test that. But it's an interesting question.
Okay, thank you.
Sure.
Hi. Were all of the cell lines from women?
Were all of them?
Yeah.
All the breast lines were.
Well, how about the pancreatic ones?
No. I don't know if they were female or not. Actually, I should check that.
Yeah, would you expect any difference in the go genes versus the stop genes?
I wouldn't expect it, but if you don't do the experiment, you don't know.
Thank you.
Yeah.
I was just wondering, and you might have mentioned this during your talk, did you at all look at the effect of aneuploidy on the rate and the likelihood of metastases in these tumors?
The rates of metastases? No, we didn't look at that. And the reason is, well, the mouse studies are just really recent. But all the data that we analyzed was all from primary tumors. So the data for the Mets isn't available, although you could say indirectly survival, of in the immunotherapy trials is linked to that, but it wasn't really analyzed. So, no.
Great talk, Steve. So I want to go back to the dark matter of the cancer genome you were talking about, where the go genes and the stop genes are basically having a dosage effect without having to be mutated, right? So have you looked at whether the go genes and stop genes also affect the transformation phenotype of cells, either alone or in combination with another driver?
You mean the ability to grow in soft auger?
That's right, yeah.
No, we haven't done that screen recently. We did it 5 or 10 years ago with the earlier version of the library, but no, we haven't done that. We've done some anoecus, but not with this library, but with specific tumor suppressor and oncogene libraries as something like that, but no.
I'm Marston Linehan. I study genetics of cancer of the kidney. And we saw, earlier today I saw 25 patients with different types of inherited forms of kidney cancer. We know of at least 18 now genes that cause kidney cancer. We've got about 40 in all 3 TCGA projects that are SMGs, but about 17 that we know drive it. I mean, we know cause it, for different reasons. So when we look at those tumors, and we were talking earlier today as we were going over our films for our patients actually about your talk this afternoon, and I told everybody in our group, you got to come to this talk. But one of the things we were sort of thinking about was we had seen, and we can look at CGH and do all that, but we've seen in many of our tumors, for example, and I've been kind of thinking about this in the back of my head, but your work and your talk today kind of maybe crystallizes a little bit of this maybe in that, so we're talking one organ, we're talking kidney. And we have these different genes of very different kinds. You know, our first gene was a VHL, that's a loss of function gene. We see that in clear cell, of course. Our second was MET, which of course is gain, and we see that. So our sense a little bit is maybe what we're seeing is haplo of these other genes, like that we consider genes that cause kidney cancer, like PTEN, like folliculin, like fumarate hydratase, succinate dehydrogenase, TSC. And that we think, we are wondering if it makes sense to think that the combination of those genes that specifically drive kidney cancer could, in an individual cancer, for example, could give us a more sort of aggressive phenotype or maybe be involved in transformation to begin with. You think that makes sense?
Yeah, I do think so. I personally, I think that, that all the sporadic tumor suppressors are haploinsufficient. Of course, you lose both alleles, it's more potent. Right. And certainly that then if you have combinations of them present in a certain tumor, that should be even, more aggressive. So I think it makes sense.
When you have, let me ask one other question. When you have, for example, high copy number, and in one of our types of tumors, we have a very high copy number of chromosome 7. Mm-hmm. And we know that the hereditary version of that cancer is mutation of a gene on 7, which happens to be MET, which is a gain-of-function gene, obviously. So, but how do you go about figuring out when you have a whole gain of a whole chromosome? So we see in the sporadic tumors, 82% have multiple copies of chromosome 7, whole chromosome. Mm-hmm. How do you figure out? So we call that dark matter because we can't figure out what's going on.
It is dark matter.
How do you go from where we are now to trying to figure out what gene it is?
Well, I think, this is a problem a lot of people have been working on. Scott Lowe has done a lot of work on this in liver cancer. And the strategy he took was either to take the genes and overproduce them to see if they give you a cancer individually, to see if they give you a cancer-consistent phenotype, or to then knock down genes along that chromosome to see if they now reduce the cancer phenotype in a mouse model. So I think that's the only way you can really prove that any individual gene is going to be driving it. But I'd also look at the focal events on that because if you do find that some cancers don't overproduce don't gain the whole chromosome, but that the focal events are there, those focal events are important in driving that whole event outside of MET.
That could give us a hint if we've got it. Okay.
Yeah.
Right. So I think I'd like to thank Steve for the very stimulating lecture.
Thank you.